Hello everyone, my name is Fernando Zhapa-Camacho and today I'm going to present our work predicting protein functions using positive-unlabeled ranking with ontology-based priors. Our work addresses the problem of protein function prediction. So we define this problem as given a protein sequence, determine its function from the gene ontology. Protein function prediction is an important task on its own, but also it serves for downstream tasks as identification of drug targets or understanding disease mechanism. From the computational point of view, protein function has been addressed as a classification task. So in this context, protein function can be framed as a classification problem because it is a multi-class problem because we have many Go functions to classify over, and also it is a multi-label because one protein can be assigned more than one Go function. However, most of the data is not labeled, which means that we know positive annotations, which means that we know that for a protein we know which function is associated to this protein, but we don't know which function is not associated with this protein. We don't know 100%. So usually this problem has been addressed as a binary classification problem, which means that unlabeled samples have been treated as negatives, assuming a data distribution like this in the picture, where the positive and the unlabeled sets are separated, and the unlabeled set is treated as a negative data. However, the actual distribution of data looks more like in the second picture, where the unlabeled set contains both positive and negative samples. Current methods adopt binary classification, which could introduce some bias into the unlabeled samples because they are treating some unlabeled as some potential samples as negative ones. So because of this limitation, positive unlabeled learning has arisen to consider the uncertainty of unlabeled samples. And this positive unlabeled learning has been applied to other contexts like system recommendations. So here we ask the following question: can we leverage positive unlabeled learning or PO learning techniques in the protein function prediction context? It's worth mentioning that PO learning has been tried before in protein function prediction, but also in other bioinformatics tasks like gene disease association or drug target interaction. And the PO learning is not only one technique, but is a set of techniques. One of these techniques is, for example, negative samples extraction, which consists on from the unlabeled set with some criteria extract some negative samples, such that the problem positive unlabeled is transformed into positive negative or the usual binary classification task. However, this type of methods usually do not get rid of the initial bias in the unlabeled set, because the technique to extract the negative samples from the unlabeled set might also have some incorporated bias. And there are also other tasks like using other type of algorithms like majorization, maximization, and so on. However, there is a third technique called empirical risk minimization, which is the one that we use and I will introduce now. So empirical risk minimization uses a decision function, or what we would call a classifier, and we are going to denote this classifier as G. So in usual binary classification task, the risk that we want to minimize can be expressed in this form, as in this first equation. So we have the positive risk, which is the risk of classifying positive samples as positives, and the negative risks, which is the risk of classifying true negatives as negatives. In this equation we have some terms like pi sub p and pi n. So pi p is some prior probability, some prior belief that a sample belongs to the positive class. And pi n is the prior belief or prior probability of a sample belonging to a negative class. These R terms can be expressed here, and I don't want to dig into these expressions. Maybe we can discuss it later, but they can be expressed as you see here. And I will mention that they introduce some loss function L that acts over the output of the classifier, and in our case we use the log sigmoid function. So how does empirical risk minimization address the positive unlabeled case? So if you notice, before we had the original risk formulation as the sum of the positive and the negative risks. However, we cannot compute the negative risk because we don't have negative samples. Therefore, some work has been done before in this area such that they re-express the negative risk as a combination of the positive and the unlabeled. So the negative risk is expressed as the risk of predicting true positives as negatives plus the risk of predicting unlabeled as negatives. Therefore, the risk, now the expression, as you can see here, is only dependent on the positive samples and the unlabeled samples and the class prior. Some other let's say tricks or adjustments have been done to this risk to ensure no negative risks. So instead, to avoid maybe this term to become too big and make this risk negative, they introduced this maximum operation here. As I said before, the empirical risk minimization acts on a decision function, which is a classifier. So we need to define what is going to be our classifier for our function prediction problem. So we use the ISM2, which has been widely used in many works, not only for protein function prediction, but other tasks involving proteins. So we use this model to obtain vector representations, and we plug an MLP classifier such that we have a ESM plus MLP classifier where the input are protein vectors and the output are going to be scores for each Go function. So if you can see here is the initial workflow, which is quite simple. We have the protein function, we have the ESM model to generate a vector, we have the MLP to generate the scores for each Go function. So in a usual binary classification setting, the pipeline will follow these red squares after the output of the MLP. The scores will follow some binary cross entropy loss and generate some predictions. However, since we are in a positive unlabeled learning, we need to compute, after the output of the MLP, we need to compute the risks. And we will only use the green pipeline for the predictions. So after the output of the MLP, we use this log sigmoid function and we compute these three risks that we need in our expression. So with this simple formulation of the pure learning, we run some experiments for the three sub-ontologies in the gene ontology: molecular function, biological process, and cellular component. So we used ESM plus MLP alone, just with the binary in the binary classification setting, and we modify this for the PU learning case, and we can notice that there is some improvement even though it's smaller, and it's not in all the cases. For example, in the cellular component case, the MLP or the binary positive negative classification works better than the positive unlabeled classification. However, in molecular function and biological process, we see some improvement. We use standard metrics in the protein function prediction task like Fmax or SMIN and AUC. And we trained 10 models with different seeds just to check that our performance is not given just by a random initialization of the models. So, in this area of PU learning, some previous work have noticed that this risk estimation would push the values either to 0 or 1. And they have identified that this might be unnecessarily hard. Why? Because positive samples should just have higher values than unlabeled samples. So it's not necessary to push the unlabeled samples to, for example, 0. But it's just okay to push the unlabeled samples to be below the positive samples. In this sense, if we take our initial expression for the PU learning risk, we take the risk for the unlabeled set, which is expressed in this way, and we modify it to be dependent also on the positive samples, such that we make sure that the unlabeled samples just rank lower than the positive samples. This is some work done previously, applied in the area of knowledge graph completion. So we are adopting this approach. So with this modification in the loss, we also notice that we could get some improvement in our case, in the protein function prediction case, and in this case we can see that the improvement is more general. It's in the 3 sub-ontologies, with the exception of the AUC in the cellular component case. Next, there is another factor in this risk minimization framework that is actually it's more important in our case, so which is the class prior estimation. As I said before, the class prior is the prior belief or the prior probability that a sample belongs to a certain class. If it's binary classification, then it's the probability of the sample to be positive. However, this prior knowledge it's usually unknown, so it needs to be somehow estimated. And there are some approaches also to estimate this class prior, and there is actually active research on how to estimate this class prior in better ways. So some of the approaches for this class prior estimation rely on instance similarity. For example, in our case it will be protein similarity, or they will do some subsampling of the distributions and try to estimate the difference or these p factors in these sub-distributions. So from the big positive and unlabeled distributions, they will sample many sub-positive and sub-unlabeled distributions, and they will try to estimate the proportions on those subsamples. However, in our case, since we are dealing with functions in the gene ontology, we decided to rely on the hierarchical information of the functions in the gene ontology. So, since protein function prediction is a multi-level classification problem, we have many classes, and because every Go function is a class, So every Go function must have a different prior, because in the previous results that I showed, this pi p was always the same for all the Go functions. However, the hierarchical nature of the Go functions, for example, Go functions very close to the root in the Go hierarchy, will be more likely to be annotated with certain proteins. So the class priors should be higher than a Go function which is very close to the leaves in the hierarchy. So following that idea, we decided to compute the priors based on the initial annotation frequency. So for example, we have the term Si, which is the quotient between Ni, which is the number of training proteins for the ith Go function, and N total is the number of total training proteins. So we have the initial frequency, and then the prior for a Go function is just its frequency divided by the maximum frequency. And we incorporated this pi o, which is a tunable hyperparameter, because some works they tune this class prior. So we decided also to this factor here to add some tunable hyperparameter just to see if this has some effect, and it had some effect. So this is the initial workflow that I showed before, and after incorporated the class priors, we obtain this. So the risks are now better informed with these hierarchical priors from the gene ontology. So after applying this class prior estimation, we also could see that the performance improved a little bit from the PU ranking case, but if you notice from the original baseline, which is MLP ESM2, there is a substantial improvement in Fmax and also in SMean and AUC. So to generate these results, we use the data from UniProt, SwissProt. We use the third version of 2023, and to generate our splits, we grouped the proteins based on similarity. So similar proteins were to a different group, and then we split the groups. We did this in order to have, in order to avoid having similar proteins in training, validation, or testing sets, such that our models do not overfit to similarity. We trained 10 models for each subontology, and then we used the trained models to test in a time split benchmark. So for the time split benchmark, we used the new annotations in the first version of 2024. You can see some statistics of the dataset here. So for the similarity-based benchmark, we tested against methods that do not use any protein similarity criteria, because we are not also using protein similarity to do the predictions. So in order to make a fair comparison, we decided to use baselines that do not use any protein similarity component in their predictions. So these methods were, apart from the naive approach, DeepGo-CNN, DeepGo-0, and the MLP, which will be our original baseline, which is not, which is our positive unlabeled learning without the positive unlabeled setting. So yeah, we can see that our positive unlabeled framework is better than the baseline. Yeah. And then, yes, for our time-based benchmark, as I said before, we generated a testing set using the first version of UniProt, SwissProt in 2024. We took the proteins that gain annotations, that had new annotations. So before, for example, other methods would consider these new annotations as negatives, but with our framework we don't consider them as negatives, but we allow for some uncertainty because they can be potentially positive. So there are like more than 4,000 new annotations, but once propagated they are more like 31,000. And in this case we did not train new models, but we use our previously trained models to predict on this dataset. And in order to compare with this benchmark, we in this case we compared again against other methods that use similarity for their predictions. Therefore, we also combined with Diamond in this case to incorporate protein similarity criteria for the predictions. So we compare against some baselines like SPROFGO, TALI, NetGo 3, AdGo Plus, and AdGo Plus. Yes, so we can see that in some cases, like cellular component and biological process, our framework is better than the baselines. However, in molecular function, AdGo Plus is much better. Well, yeah. So in order to dig a bit more on this dataset, we decided to perform a term-centric evaluation. So we decided to compute the AUC for each Go term. So why we did this? Because we had the intuition that since we are using the frequency as the Go functions annotation frequency as the class prior, our method should perform better in Go functions with higher specificity, which means that it will be Go functions with the fewer number of annotations. That's why we decided to compute the AUC for each Go term, and then we group the AUCs by the number of annotations. And you can see that in the case with a lower number of annotations, the PU learning setting, which is the blue dot, are better than the MLP, and they are a lot better than the AdGo. We decided to compare with AdGo because previously you could see that AdGo also had good results. However, you can see that AdGo is better in some cases or comparable to the PU learning and the MLP for Go functions with very large number of annotations or very general functions or functions with lower specificity. But for functions with higher specificity, MLP and PU learning perform a bit better. So that's our results. We have some discussion points like the classification model. So we used relatively simple classification model because we used ESM2 plus an MLP. However, some baselines that we use, like that we consider, like NetGo 3 for example, uses other type of information like sequence homology or literature. So our PU learning framework could also be extended with, let's say, better classifiers or classifiers that incorporate additional information, not only the protein sequence. But a more interesting discussion point is also the class prior estimation. So as I said before, most approaches rely on statistical or similarity aspects of the data. So in our case, instance similarity would be protein sequence similarity, for example. In our case, we use the the Go hierarchical structure to compute the class prior, but still it is more like a class prior estimation from the information point of view. However, this case, the protein function case, allows for some input of biological information for the class prior estimation. For example, we could incorporate the type of organism in the class prior estimation, because if we have functions like anatomical structure development and we are predicting for bacteria, then the prior for this specific function will be 0. Or in other case, or other example, we could take the environment in which the organism was found as a source of class priors. Because if we are using this function, for example, swimming, then the prior, if we are predicting for functions of organisms in the sea, then the prior will be 1. So there is like this type of information that can be included for potential future work. So to sum up, what we did in this work was mainly to adapt the PO learning framework in the context of function prediction, leveraging some work in other contexts like knowledge graph completion. We adapted also the class prior estimation based on Go hierarchy only, although more other type of information, like, as I said, biological information could be also used in the future. We showed that PO learning can improve over standard binary classification in this task, and we believe that there is some potential future work because the PO learning research is not also based on positive unlabeled learning, but there is also some work on positive unlabeled negative learning. So in the case that there exist negative samples, this framework has also been developed. So if we manage to get negative samples for this case, the protein function prediction problem, we could also extend the framework to include these negative samples. We could generate these negative samples with different strategies, and one of those could be from background knowledge using the gene ontology. Maybe use entailed negatives, logically entailed negatives, and also this problem can be framed as semantic entailment. Also, as I said before in the discussion, we could use more sophisticated classifiers, which not only consider protein sequence but also literature or sequence homology, and yes, incorporate biological information in the class prior estimation. So I think this is it. I thank you.
Hello, thank you. That was a fantastic talk. I want to ask, when you were looking at your performance on terms based off of the size of the terms, and you found that your PU-Go method was doing significant better than the multi-layer perceptron, do you know if it was doing similarly well on molecular function, cellular components, and biological processes in each of those on the terms that had a low number of annotations, or did you see a divergence in the between molecular function, cellular component, and biological process?
Was online at the beginning of the talk. Well, this one. Is he on it? Something like that. No? He's not. Yeah. We just proceed. So then I'm not sure. Then it's gone.
I can send him an email.
He was thrown out of the system, he says. Logging back in. Yes, but he can't find. He says he got logged out. He's coming back. Yeah. If we still have time for him. Yeah. I don't know.
Any other questions?
Well, only if he can actually answer.
Yeah. Well, I saw some of the co-authors on one side of the phone online. I don't know if they want to try and take a stab at answering some of these questions.
But I think I can maybe answer this, but I don't think the analysis for the different sub-ontologies is available. Fernando might have the data. I haven't seen it.
Okay.
So he might have generated the data, but he may have just generated it and not split it by sub-ontology.
Yeah, it could be that, they were all the same, it's not worth showing this for plots, but I was just curious.
Yeah. Is he back online? I don't know. How do we see this? Just to make sure, just to check if he's there.
These are the attendees.
Next
stage is
Okay. That did not work very well either.
But if you're willing to answer more questions, I had one.
Okay, I can try my best since it's yes.
On the spot. All right. So one of the things that I was wondering is how do you see this might be working on non-model organisms? Because it seemed a lot like this is relying on a lot of the existing data sets. And when you throw something really new and unknown to it, how do you think it will perform?
Okay, so the question is how this would work on model organisms, non-model organisms. Yes. Okay, so maybe. So Fernando?
Yes, can you hear me?
Yes.
Yes, sorry.
So maybe we can not see you, but we can hear you. So the question is, excellent. So the question that Hande asked is how this would work on non-model organisms.
I will need to ask, what does it mean, a non-model organism?
Mm-hmm.
Yeah, well, usually non-model organisms. So it seemed to me like your data was really relying on very well-known data, or at least well-known organismal data. So I'm wondering if you were to throw something that's not really non-model, so not well studied, how do you think it will perform?
Okay, so yeah, well, for this work, we didn't actually use any because we are doing this positive unlabeled learning, so trying to process the unlabeled set using some prior information. But in this work, we didn't rely on biological information for this. We only relied on, let's say, the hierarchy of the Go to introduce prior information to the unlabeled data. So I think it can still work for the data that you mentioned, as long as you can input these prior probabilities or prior beliefs to the Go functions based on some known frequency of these Go functions.
Hello, I actually two questions. So one is I asked this, just want to confirm. So when you are predicting, comparing the multi-layer perceptron to your PU Go, yeah, thank you, Simon. Sorry, I'm a bit tall. The microphone was low. When you're comparing your multi-layer perceptron to the two other, your PUGO and the third method, depending on the size of number of proteins annotated to each term, and you found that the PUGO was doing a bit better than the multi-layer perceptron across the board and much better than the third method. Did you see divergence on those lower terms between the three subtrees of Go, of molecular function, biological process, or cellular component? Or in that low number of annotations area, were they pretty consistent?
Yeah, well, actually, we didn't analyze by sub-ontology. We just analyzed by the number of annotations to each Go. But yeah, that's a interesting question. Yeah, but we didn't analyze in that way. So I do not have an answer for that.
Thank you. And my
Yeah.
second question was, you had talked about in future work the potential of adding in negative labeled data into this model in addition to the unlabeled data. And I was wondering if you had any ideas on how you're going to approach that, because with the gene ontology, if you're still using that as a base, it seems like we only have information about positive labeled. If the gene ontology doesn't contain information of, no, this gene doesn't do that process. So I was wondering if you had any idea or any insights into how you can find this negative label data in the future.
Yes, so one potential idea to obtain negatives is using the gene ontology axioms. So deductively apply deductive reasoning to generate some entailed negatives. So this will be negative annotations that come from the theory that is built in the gene ontology. That will be like one way to generate some negatives. Yeah, because the number of negatives that currently exist are very little. So yeah, we need to generate some more, and that may be one way to do it.
Thank you.